Hello, good evening. Uh, can you hear me? Hello, Genevieve. Let me know if you can hear me well. Hope you all are doing fine on a Friday evening. Hello, Barbara. Okay, okay, good. So then I think I'll start in 1 minute. Uh, yeah, I mean, hopefully it will be like a 1 hour, hour and a half session today. But let's see how fast we can how fast we can go. Because a lot of steps take time, but we won't wait for all the steps to finish. Yeah. Okay, but I think we can start. Yeah. So, welcome again, and to this new course on next generation sequence analysis. Uh, so it will be conducted by Yura Song and myself, and uh, we will have the course will be divided into 3 sessions as of now. Today we will just talk on introduction, so on the different file types and next generation sequencing, on how to do some kind of mapping. Then the next session is in 2 weeks. It's on a Thursday, at least for now, and it will be on we'll do the RNA-seq analysis. And the last thing, in the last session, we will perform ATAC-seq analysis. So for both session 2 and session 3, we'll upload the files, which you will need to map, just like we will do today, before the session starts. So there'll be a little bit of preparation for the next 2 sessions. Yeah. And as always, the course details are on the GitHub repository. So all the details are there, all the details about the session, the YouTube videos, everything. The preparation, the data is there. And you will, whenever there is anything that you want to look at, just go to the GitHub website. Yeah, this will be updated continuously. Another thing is that if you have some issues, you are more than welcome to email either myself or Yura or both. It's no problem. But it would be better if you also post your issue on the GitHub website. So if you see here, oh, actually it's cropped out here, but there is an issue button on top. So if you have any issue, then you can just put here. For example, a couple of people told me that on Windows, FileZilla has some malware. So it comes with some kind of virus if you try to download and install it on Windows. And so this was something that they communicated, and it is a problem. So that's why I think now we suggest to use CyberDuck. So this will be really nice, because in this way we keep a record for every course, whatever the issues have been, and this would help us in future. Yeah. So just if you want, you can do both. You can post the issue on GitHub page and also email. But if you post it here, we will get an email. So don't worry about it. We will reply to it. Yeah. So just it would be better to make a record of all the issues that you guys face during the course. Yeah. So what is the course about? It is about doing next generation sequencing analysis, but using Galaxy. Yeah. So this will be our tool of interest. It's a very powerful tool. So it should be able to do everything that you want. Yeah. So this would be something that we'll be using. And the aim of the course is that after this course, you can go from raw data to results. not via analysis, of course. Yeah. So that you're not really dependent on anybody else to do any steps for you. So once you get the raw data from your sequencing platform or from whenever you have submitted your samples for sequencing, you should be able to do all the steps. At least get like differentially expressed genes or differentially expressed regions for ATAC-seq, along with the p-values or statistical significance. So at least the basic pipeline you should be able to do by yourself. Yeah. And so that is why we will go through all the steps and the tools in this analysis pipeline. It should allow you to work independently with your NGS data without getting any support from bioinformaticians. Um, and you should also be, at the end of the course, be able to develop Galaxy workflows, which you save. And then, you can just run them. You can automate the process. So you don't have to redo all the steps again and again. Once they are done, then they are saved as workflows, and then you can automate. So you go much faster. So this would be the aim of the course. But the things that the course will not cover is that because we restrict ourselves to using Galaxy, so it will of course not cover tools or pipelines which are not present in Galaxy. So if there are things that people have implemented but that Galaxy has not imported, we will not be able to cover that. We will also not cover how to execute these tools in shell script or on a cluster. Although we are thinking of having a session on this after the course, so how to actually do these things in shell script, but that may be in a very small quantity and not live, because we would have to do that in person. It requires a lot of installation and a lot of debugging. So we'll possibly do this on a day, like we'll have like a maybe 6-7 hour session on a single day and cover it in person. But this also requires prior knowledge of coding in shell script, so it will be a bit more restricted. But anyway, the current course doesn't cover that, so we are restricting ourselves to Galaxy. So of course, if a feature is not implemented in Galaxy, we will not be able to cover it. And also, Galaxy is very good with working with model organisms, for which, for example, the genome and transcriptome is well known. Technically, you can also work with non-model organisms, but it's a bit more of an effort, and we will not be able to go into too much detail. So, if you're working with things like zebrafish, mouse, human, Drosophila, C. elegans, you'll be able to do things very quickly with Galaxy. Yeah? And last thing is that we will only be focusing on bulk RNA-seq and also bulk ATAC-seq. So not single cell. Yeah. So we will leave single cell either for another course or later. Yeah. But this one will only be about bulk. Yeah. So let's just dig into it. So we'll start today with the mapping of data from RNA-seq. So you have given your samples for RNA sequencing. They go into a machine. Yeah, most of the people don't really see it, right? And then you get a big file back, like a few GB file back, right? Then what do you do with that big file, right? Most of the time, you ask somebody to give you counts data, not the raw data, right? But today we will deal with raw data, right? So, but first, before going into what the raw data is, we'll briefly go into the steps um, that the steps that you go into while making the library for sequencing. So what actually goes into a sequencing run? So you have your total RNA that's in your cell or your tissue or however you have collected it, right? And this is one of the pipeline for making libraries for RNA-seq. There are many different ones, but it sort of, in the general format, it's the same. The end product is more or less the same. Yeah. So in this pipeline, what they have done is they have fragmented the RNA. So they chop it into smaller pieces. Then to sequence, of course, it's DNA sequencing, right? I mean, it's Sanger sequencing at the end of the day. But so that's why the RNA has to be converted into some kind of DNA molecule, and this is done by reverse transcription. So in the fragmented DNA, you can add random primers. Yeah. So in this case, it's also important to remove ribosomal sequences, and this is done by using magnetic beads, which have complementary sequences to ribosomal DNA, to ribosomal RNA, and that pulls them down, right? Otherwise, if you want to enrich for RNA, then another way is to use a poly T primer here above without fragmentation, and then fragment later. Yeah, so it depends what your protocol is. But in this case, you first remove the ribosome, fragment the RNA, then use random hexamers. But the most important thing in all of this is what happens in the last step. So in the last step, the cDNA that has been generated gets attached with something that's called the barcode and the Illumina index. Yeah, this is the most important thing, because this is the thing that allows you to, first of all, get your sequencing reaction, and second of all, it allows you to know which of the RNA molecules or which of the cDNA molecules belongs to a particular sample. Yeah. So Illumina calls them I5 and I7. Yeah. And these are the barcodes. So these are unique for each sample. So depending upon the combination of I5 and I7, you will tag each sample separately, right? And that also means that in a single reaction, you cannot really reuse your I5 and I7. So this combination has to be unique for every sample. Yeah? But in addition, you have this universal tail. Yeah, and we will go into why you need this universal tail, right? But this is the important thing. Your cDNA is not naked cDNA. When you do next generation sequencing, it has adapters or some extra sequences added to the ends, right? This is important because this is also something that when you get your results of RNA-seq data, these sequences are added to the ends, right? So here is just a small video.
That consists of sequential blunt-ended adapter ligation, 3' adapter, then 5' adapter, to total RNA. The adapter ligated RNAs are then subject to RT-PCR to enrich for RNAs with adapters in the correct orientation. Products of RT-PCR are run on an agarose gel, and the desired products are isolated at 147 and 157 base pairs. The purified library is only validated qualitatively using the bioanalyzer.
Yeah. So that is how you choose. I hope that the audio and the video, the audio was like you could hear the audio from here. Yeah. So you can see that you have put the adapters, and you have chosen a specific fragment for sequencing. Yeah. So there are two things that have been done: adapter addition and the choice of specific fragments. Yeah. So at the end of the day, you will either have three kinds of cDNA molecules. The most common one is the unique dual index. Indexing is what is the addition of barcode so that you can separate cDNA molecules between samples. Yeah, that is what is called indexing. And dual indexing means you add an index on both the sides. Yeah, dual indexing does not mean that you do a paired end sequencing. Yeah, that will come to later. Yeah, you can do dual indexing but a single end sequencing. But dual indexing just means that you have added a barcode on both the sides of cDNA. Yeah, that is dual indexing. So the combination of I5 and I7 provides you with the sample information. Next, you have this universal end, which is the flow cell binding sequence. Now, why do you need a flow cell binding sequence here? Yeah. What is the flow cell? Flow cell is this glass, is a chamber which Illumina uses for their sequencing. And I'll just show you a video explaining this phenomenon.
Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell.
So just to make things clear, this universal sequence at the end is needed for the first hybridization to this cluster. Yeah? Without that, you wouldn't really hybridize it. Yeah?
Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over, and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The three prime ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template.
So just to explain this also a bit more, so why clustering is important is because this is not single molecule imaging. So if you just have one molecule of cDNA, the signal that it makes is not strong enough to be detected. So instead, what you do is you have these clusters, and in each cluster, a single cDNA is captured, which is then amplified millions of times. Yeah, so you get the same sequence amplified a lot. Then when a nucleotide gets added, it's actually added to that millions of sequences together, right? And then the signal that it generates is strong enough for a camera to capture it. And because the whole cluster has exactly the same sequence, more or less, you get this bright spot related to the nucleotide. This is different from Sanger sequencing because Sanger sequencing requires it requires the termination of polymerase, right? So you put dideoxynucleotides, right? And as soon as a dideoxynucleotide is incorporated into cDNA or into DNA, it terminates, and then depending on the length, you know what the sequences are. So that's Sanger sequencing. So this is different than Sanger sequencing because here what you are detecting is the fluorophore that is released upon addition of a nucleotide simultaneously to millions of molecules. Yeah, so that is what is being detected here.
The addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read.
So index 1 is the barcode, right? So this would tell you what sample was. It will tell you from which sample did the cDNA molecule come that is being sequenced in a particular cluster. So that's what index 1 is doing.
After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step.
Yeah. So index 1 and index 2 together uniquely define a sample. Yeah.
Clustering is a process wherein, in this step, extend the second flow cell oligo, forming a double to read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving.
So this is now the difference between single-end sequencing and paired-end sequencing. So right now, what have we sequenced? We have sequenced one side of your cDNA, right? Typically 250 base. I mean, depends on the flow cell, but it varies from 25 base pairs to 250 base pairs, right? So we have sequenced one end of your cDNA. We have sequenced index 1. We have sequenced index 2. Now, if you stop your sequencing reaction at this stage, this is called single-end sequencing. So you have sequenced your sample information, the sample barcodes, and one side of your cDNA. In paired-end sequencing, you make this bridge, and then you sequence again, but this time you sequence the other side of cDNA, right? So in paired end, you get both the sides of cDNA that were ligated with adapters. Yeah. So this is the difference between single end and paired end. Single end sequencing does not mean that you cannot do dual indexing. Yeah, you can do dual indexing with single end. Similarly, you can do single indexing with paired end. If you do not have so many samples, you do not need so many barcodes. But more the barcodes, the more samples you can sequence together. Yeah, I hope that makes sense.
Reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification.
Yeah.
The paired end information is used to resolve ambiguous alignments.
Right. So in the last, he said that the paired end sequences are used to resolve ambiguous alignments. We'll go into that. Yeah. So the last 2 sentences that he said, we'll look into it later. Yeah. So the question is, why do you want to sequence many samples together? Why not just sequence one sample independently? And that's because the capacity of a single flow cell is very big. So currently, like if you look at NovaSeq, there are different machines. NovaSeq is a kind of machine. HiSeq is a kind of machine from Illumina, and then there are different versions of it. So if you see in this machine, in a single flow cell, you can sequence 8 to 10 billion. You will get 8 to 10 billion DNA base pairs, right, per flow cell if you do single end. And if you do pair end, then of course it gets doubled, right? So another thing is, if you did single end chemistry, it does not mean you will get double the DNA, because your number of chambers still remain the same. So each chamber has to give you the read from the single end, right? So the paired end read will just double from single end, right? So just doing single end doesn't give you more, doesn't allow you to have more samples. Yeah? And typically for one sample, it's a good starting to start with 10 million reads. Yeah, and sometimes you would want to go higher, and we'll discuss why you would want to go higher. So, for example, if your sample comes from very low amount of RNA, then maybe what you sequence over time is just duplicate. So you just keep on sequencing the same molecule over and over and over again, right? You do not get more complexity. Yeah. So after a certain period of time, you saturate, right? So, but and also you can run the same sample again, right? So you can ask your provider to first give you 10 million reads. You look at how your sequencing went, whether it was good, whether it was good enough to get some data out of it, and then you can ask them to sequence 40 million more or 90 million more, depending on how much saturation have you reached, right? So because for each sample 50 million is kind of enough for all the experiments, when a flow cell is giving you 8 to 10 billion, you would want to combine as many samples as you can, right? And that's why you need all this indexing. So you will not be the one who will be demultiplexing the data that comes out of the machine. So that is the responsibility of the person who is doing the sequencing for you. That's already an Illumina provides them with that information, right? So you will not be separating the samples, but what you will get is information about your own sample, right? You will get a file which is called the FASTQ file, yeah, which will be your raw data. This will have all the sequences related to your data. So what does it mean to your data? Related to the barcode which was used to prepare your samples. And of course, while preparing the samples, you need to know which sample got which barcode. Yeah, I mean, not you, but if somebody else is preparing, then they would have to keep note. But yeah, that information has to be kept track of. But you will get information of your sequencing run with most possibly the index and the adapter sequences in your raw data, right? Because this data is sequenced, so it is part of your data. It is part of your file as well, right? So this is something that we have to remove. Yeah. But anyhow, first, how will your data look like? So it will be in the FASTQ format, possibly a few GB. Yeah. And what is the FASTQ format? It's the FASTA format, which is the simple format to store DNA sequences, but with additional quality scores. So that's why it's FASTQ, yeah, FASTA with quality scores. And I think most of you must be familiar with what is a FASTA format. So it has two parts: a header, which has the name, mostly the name of the sequence, and this header is followed by the sequence. So mostly it's ATGC, but it can also have Ns and other combinations of nucleotides, but in most of the cases it's just ATGC, right? Now what are these quality scores? Oh, I think that got mixed up, but anyhow, so if you have done Sanger sequencing, then you would know you would get a chromatogram of something like this, right? And here underneath you see the quality scores. So you see here is the quality score. So if the quality score is green, above 30, that means that whatever the algorithm is, it's very confident that whatever nucleotide is being called is the actual nucleotide at that position. So for example, in this stretch AAT, it's very confident that this is actual nucleotide, right? However, if the sequencing, the quality score is a bit lower, that means that there is less and less confidence in what was done. So for example, if you look here, then at this position, you actually see two peaks, right? A green peak and a red peak. So even though it calls A, because A corresponds to this green peak, which is higher, it's not really sure. And that's why it has reduced the quality score. When you do Illumina sequencing, your sequencing is, I think, close to 80% accurate. And that is because your single molecule of DNA is amplified millions of times. So if a base pair was mutated at a very early stage, you can imagine that half of the sequences have a certain base pair and the other half have a different base pair, right? You get this sort of lineage effect, clonal effect, right? uh, and then at that base pair your quality score will decrease because it will look as if you have two different base pairs, right? And this is what the quality scores suggest to you. Yeah. um, if you compare this with single molecule sequencing, for example Oxford Nanopore, there the quality score is very high. Sorry, the the missing base pairs or the wrong base pairs are very low. Yeah, so the quality source is mostly very high. So I think its accuracy is close to like 95, more than 95%, right, in the single molecule sequencing. But with Illumina, it's I think close to 80-85%. And this information is conveyed to you in this FASTQ format, which is essentially the FASTA format with the quality scores added. So how does the FASTQ format look? It has a header also, which here is called label, and it starts with @. So here, most of the time you have information about the kind of Illumina machine that was used and whether it's paired end or single end and so on and so on. Yeah. Then it's followed by the sequence, the DNA sequence, followed by a plus sign. So this plus sign signifies that the DNA sequence has ended, and then the quality scores have begun. So these quality scores are called Q scores, and they are in ASCII characters. I'll come to that. So for every base pair, you will have a corresponding quality score. So this means that this base pair C has a quality score of A, and so on and so on. So like, for example, this T has a quality score of colon. Now it doesn't really make sense, right? What does this A colon 9, all of this mean? So they are in ASCII, in which this is the lookup table. So it's again a score out of 42, yeah, with the corresponding p-value. So for example, A is a score of 32. It's pretty high. Yeah, so it's a very good score. So there is very high probability. There is very low probability that there was an error. That there was an error in calling that base pair. Yeah, so most possibly this base is correct, right? And that is why you see the A here. So that this is score 32, 32, 32. Then colon. So colon is 25, right? 9, the number 9 is 24, and so on and so on, right? @ is 31. Let's see if there are only some bad base pairs. Question mark. Okay. What is a question mark? It's also 30. So it looks like very high quality, right? So this is how the FASTQ file looks like, right? Let's look at the FASTQ file that you are going to use today. So before we look at that file, we will look at it in Galaxy. But just to introduce to you, I did not put a slide. So the FASTQ file that I have given you is of RNA-seq data from zebrafish beta cell. Yeah, and you will see that it's only a few kilobase. That's because it only has 1,000 reads. So initially it had millions of reads, but I truncated it to only having 1,000 reads, just so that we don't take too much time today analyzing it. Yeah, and so I will be using the Galaxy with you, live coding it. But these things do take a bit of time. So it is possible that while Galaxy is working, we talk about something else or even proceed to the next step. And then once it finishes for you, then you can have a look at the result. Yeah. So let's look at the file. Okay, let's go to the Galaxy, and then first we'll start with Galaxy. So you can go to your web browser and access the Galaxy server that is closest to your region. So if you are in Europe, you will use galaxy.eu. If you are in US, use galaxy.org, and if you are in Australia, use galaxy.org.au. Yeah? That is also on the GitHub. Yeah? And I hope that most of you have registered, all of you. If not, technically, you can also use it, I think, without registering. but then you cannot really, I think you cannot remember your history and all the steps that you did. So I would suggest to register. Yeah. So if you have registered, then you will see that on the left hand side here are the tools for analyzing your data. So these are all the tools, all the steps. Yeah. In the middle is actually a viewer, and if you perform any step here in the middle, you will see your output. Right? And on the right hand side is your history, and this history will also contain the data sets that you have uploaded and all that kind of thing. Yeah. So first we will log in. So we press that login. Again, I do not know how it will work in live coding, because a lot of internet is being used also for streaming. So if things become slow, then just excuse me. Yeah. So for login, you use your email ID and the password that you have, right? And then you can go in. Right? As you will see, nothing has changed. But now, because you have logged in, your history will be remembered, yeah, for the next time. That is one thing. And second thing is, if you upload your data via an FTP server, and we'll come to that in like a couple of minutes, then you can access it after logging, right? So I hope you have downloaded the FASTQ file from GitHub. If not, then I'll just show you where it is very quickly. So you go to data and download this FASTQ file. Yeah, and it's only a few kilobases. So it's only the first 1,000 reads. Yeah, and it's single length, so it's not paired in, because we'll do paired in next session. So if your sequencing is single end, then you only get one FASTQ file, right? Because it only has the data from one end of your cDNA. And if it's paired, then you get two FASTQ files, which are coupled to each other. So one file will have information of one side and another file from another side. Yeah? So this is single end FASTQ file. So the first thing is, well, you want to get your data into Galaxy. So you just say get data. Yeah. So anything that you want to do, they have merged it into big groups. Yeah. So most of these are self-explanatory. So you get data. Now there are many options on how you can get data into Galaxy. If it's a publicly available data set, then you can get it directly from GEO, so Gene Expression Repository from NCBI. But here we want to upload our own data. So to upload your own data, you have this option, which is upload file from your computer. Yeah. If you click on it, you can choose a local file. Yes. And we will do that. So wherever you have saved your FASTQ file, you can go to that directory and choose it. Yes. And if you press start, then it will start the upload. So if you do not have an FTP server, an FTP client installed for you, then do this, because it's only a few kilobase. It's fine. But I will also show you how to do it with an FTP server. And for that, I will be using, so I will not be doing it this way. Instead, I will be using an FTP server. Yeah. So what is an FTP server, FTP client? So the websites used an algorithm for communicating with each other, a protocol which is called HTTPS, right? The World Wide Web algorithm, right? FTP is another handshake or another program to communicate between two computers. Yeah, and it's mostly used for file transfer. It's a faster way of communicating files with each other. So Galaxy has dedicated FTP servers where they store their files. And to connect to that FTP server, you will need an FTP client. So FTP client is just a software which is on your computer, which can connect to an FTP server. So just like the web browser connects to the internet via HTTP, FTP clients connect to FTP servers, right? So it's exactly the same thing. Yeah. Although, yeah, anyway. So which are the FTP servers? So it will depend on which kind of Galaxy you are using. Yeah, and if you want, you can. I'll keep it here. So if you are using the .org, then your FTP server is use galaxy.org. If you are using .eu, then it's this one, galaxy.uni-freiburg.de. Yeah, and if you are using .au, then use galaxy.org.au. Now let's connect to it. So we will be using CyberDuck. It's working well, but you can use any other client that you want. So just like when we want to go to a website, we have to put the website name, right? Here, similarly, whenever we want to connect to FTP, I'll put this on the side so you guys can still see it. Yeah. Whenever we want to connect to an FTP, we have to put the name of the FTP server. It's as simple as that, just like connecting to a website. So for CyberDuck, it will depend on what kind of client you are using. But for CyberDuck, you can say connect to a server. Yeah. And this thing should pop up. Or in any other FTP client, you only have to specify 3 names: the address of the server, the username, and the password. Right. So the address of the server will be depending on which kind of Galaxy you are using. And in at least CyberDuck, it adds FTP colon 2 forward slashes to the server name. If your FTP client doesn't add, then you have to add it. Just like a website, like when you look at a website, its address has HTTPS, right? Colon 2 forward slashes. Similarly, the FTP client or FTP servers have FTP colon forward slash in their address. Yeah. So we will go to Galaxy dot. So I will be connecting to the EU one, Cyber dot day. The username is the same username that you had for when you logged into Galaxy, and that is why it's good to register. If you just registered, give it a couple of hours before accessing the FTP, because sometimes it takes time to sync. Yeah, so maybe you cannot access the FTP for a couple of hours after registering. Yeah, so this will be your email address, and I had my password saved, so it just comes. Yeah, and there will be no private key. Port will be 21. Yeah, so these will be the things that you have to put, right? So just make sure that FTP double colon forward slashes is added. If not, then you add it. Yeah, and then connect. And when you connect, in here you will see a directory, and this directory has all the files that you have uploaded. Yeah. So if you want to upload a new file, then you make a right click. For CyberDuck, it's a right click. For different ones, it's different, right? So you make a right click and you say upload. Yeah. And I will upload this FastQ file. It's actually already uploaded here, so I will just overwrite it. And that's it. Now, once you have uploaded, if you go back to Galaxy and you go to upload file from your computer, then you do not have to upload it from your local file. You just say choose FTP file, and it will show you your FTP files. Just one more thing to add, it actually says to connect to this FTP server, use galaxy.eu, and that's completely fine. You can do it. Just the Freiburg one works faster, and it's also more stable for some reason. Yeah? So you can also use galaxy.eu to access this one. So whichever file you want to upload, for now, you say start. Right? And then you will see that this file comes in your history. Now, it has this clock button in front, which means that it has not it has become part of a queue on the Galaxy server. The whole command has not completed its execution, and you can also see here. So how the Galaxy will work is you submit a command to execute, it will become part of the queue, and when it starts getting executed, it turns yellow. That means this is getting executed right now. The command that we ran was upload data to Galaxy, right? This is the command that we ran. And now it's getting executed, and after a bit of time it will turn green, and green means that it has successfully uploaded. Yeah. So you can see that a few kilobase also takes time. It's normal. Yeah. But the FTP transfers are much more faster when you have gigabase of files, especially when you have multiple files. So let's say if you did an experiment with paired end sequencing and two conditions with 3 replicates, then it will be like 12 files, each of few gigabytes. Yeah. So it will take, the FTP will be much faster. So once it's executed, you see it has turned green. That means the execution, first of all, the execution occurred. Second of all, it occurred successfully. If it was unsuccessful, it would turn red. Yeah. So red is unsuccessful executed, green is successfully executed. So gray with the clock means in the queue, yellow means currently running, green successful, red unsuccessful. And now here it starts to give you more option. First of all, it already directly detects what kind of sequencing it is. So it calls it FastQ Sanger. That's just a different way of calling FastQ. Yeah. And it's also zipped using gunzip, and that's why it is .gz. So .gz is just a compression algorithm. It's called gunzip. So like a zip is a compression algorithm. Similarly, gunzip is another compression algorithm. Yeah. And you can download. Yeah. So anytime anything is in your history, you can download. First of all, you can also name your history. So that would be good to name now. So let's name it June 5th. And let's name it single end RNA zebrafish. Yeah. So you know what is what. Oh, why did it not work? Okay. Yeah, you have to press enter. If you just press somewhere else, it doesn't do it. So change the name and then press enter. Yeah. So you can get information about the command that you ran. So this was the command that you ran. You use the Galaxy tool, which was called upload, right? And all of this is more metadata that most of the time you're not really interested in. And this was what was run on the shell script. Yeah. So if you are doing it on your own local computer, then these are the kind of commands you will be running. Yeah. And these are just more information. You don't need it. The more important thing is this I, which is view data, which is really, really important, right? So when you click on this I, and if it's a human readable format, this is important. If the format is human readable, then it will show you some part of your data, at least the top. And again, here you'll see that this looks like a FASTQ format. So you have @, right? This is the header. This is some information about it. Yeah. I think I also do not know how to read this information, but it's something to do with Illumina and everything. I think this thing is repeated. Yeah. So from here you can get information about where it was sequenced, how it was sequenced, and all that kind of thing. Then this is the sequence. So this is the first cDNA molecule that is present here. The second one, the third, the fourth, right? Now every sequence is followed by a plus. That means that the sequence has ended, and then the quality scores begin. And as many sequences you will have, those many quality scores you will have. So for each sequence, there is a corresponding quality score, right? So this C was sequenced with the quality score of @. So what is @? At the rate is 31, so that's pretty good, right? Let's see if there are something bad. So bad is this thing. Yes, so you see this bracket, right? That is a quality score of 8. So that's not good, right? So there is very less confidence that this base pair is correct. Yeah. So this is how the actual data. This is raw data. This is what we call raw data, right? This is what you will be getting from your sequencing platform, right? And this is the way to upload it into Galaxy. Now we will start at least some kind of analysis. So the first analysis that we will do is to just have a look at the overall quality of our sequencing run, not the quality of each molecule of each sequenced read, but the overall quality. And for this, the tool to use is called FastQC. You can search for it here. Yeah. So FastQC is a quality control, a quality reports for FASTA. Yeah. So you click on this tool, and certain characteristics come up, certain parameters come up. One thing that you can do is, if you are going to use a tool many times, you can click on this star here, and then it becomes one of your favorite tools. Then in future, whenever you want to use it, you don't have to search for it. Just go here, show favorites, and within the favorites, you have saved everything, right? So what does FastQC need? First of all, before what does it need, it is good to go down, to just scroll down and read what FastQC does. So most of the time, the description of the tool is given here, or if you have run a tool, it's given with a question mark here. So that is if you have run the tool, right? Otherwise, you can read the description of the tool here. So the FastQC, it aims to provide a simple way to quality checks on raw sequencing data, right? And it gives you a quick impression whether your data has some problem, right? So it will look for certain biases in base pair, right? Because if you have sequenced millions of read, then most of the time at each length, the probability of getting ATGC should be more or less 25%, right? It will check for adapter sequences, it will check for very short sequences, and so on and so on. Yeah. So it will give you an impression of your whole sequencing run. So what are the input parameters? It needs some kind of data, right? So we have already uploaded the data. So that is what we can put as input. If nothing is showing up, you can click on browse datasets, and browse datasets will allow you to choose data. Yeah. So if you have imported many different FASTQ files and you only want to check one, then you can choose which one to check. Yeah. Then you can upload more things, but in this case, we don't have anything else, right? So you can always ask your sequencing platform if these kind of parameters, if there is some kind of effect, some kind of special effect for your sequencing run. But for normal sequencing reactions, you don't have to specify anything else. Yeah? You don't have to specify adapters, because this tool FastQC already knows about Illumina adapters. Yeah. And so don't need to change anything. You just execute, right? And when you execute, you again see it has entered into the queue. So it's gray in color, right? And if you click on one of them, and you can click info, right? And it will show you how it was run. So what were the input parameters? So you can always go back and check it, like, if the input parameters were correct or not. So the input parameter was to have the short read from here. Yeah. And for me, it has started to execute. So it becomes yellow, right? And it will execute in a little while, right? So until then, we can go forward. So this was the connection to the FTP server, right? So it has finished its execution, so it's pretty fast, right? And you will see that one of the output is HTML because it gives you a report in HTML format, and another output is in text format. So the text format is just essentially the HTML format in text way. So the text one is more interesting. So we will say view data, yeah. And when we view the data, let me just zoom out a little bit, yeah. So this is all the different reports or summaries that FastQC has made. So it has made some basic statistics. So what was the, how many sequences were there and such sort of things. Yeah. It has made a mean of the sequencing length. So I think this sequencing was 75 base pair, right? So that's why it's around about 76 base pair, and of GC content. So okay, 43%, right? So just remember that I only chose randomly like the first 1,000 reads. So that's why a few of these summaries would not look perfect, yeah, because it's not millions of reads, yeah. And then next, it shows your quality score on average per base pair. Now, as you can see, in green is good, right? Yellow is okay, and red is bad, right? So on average, the quality score, at least for the first 35 base pairs, is good. Now, of course, the more away you go from the start, the quality goes down. Yeah, and then also some of the reads reach bad quality, right? So this is very common. This is how your all the data looks like. Initially, it's all in the good range. It should be. If initially itself your scores are in the middle or even in the bad range, that means something went wrong with the sequencing. Most possibly, something went wrong with the flow chamber. Yeah. But with the length, the quality score generally degrades. Yeah, that's completely fine because we'll take care of it in the next step. So it's nothing to worry about. Yeah. Um, then it will tell you what is the quality per tile. So on average, what are the quality scores? It's okay. Yeah. Um, then it will show you the distribution. Let me see if I can hide this thing. Yeah, I doesn't see. Yes, here. Yeah. Okay. Perfect. So on average, what are the quality scores, right? So that means that um, on average, at least quality score distribution over all sequences per sequence. Okay. So what this means is that at base pair 35, what is the average of the quality score, right? So on average, it's very good quality until 30. Well, this is the quality score and this is the length. Okay, it's my mistake. So this means that if for a particular length of sequence represented on the y-axis, what is the mean quality score? So it's opposite. Sorry, my mistake. I was reading it in the other way. Yeah. So um, yes, score distribution over all sequences. Okay. So on average, this is difficult to understand. Okay. Anyway, we'll move ahead. I'll look at it, what it exactly means later. But this is a more important one. Per base sequence content. This is something that what it shows is at a given base pair, what is the percentage of ATGC, so of the particular nucleotide. So if you have a lot of reads, typically after 12 to 13 base pair, it averages out. Initially, it does not average out because you have your indexing, because you have your adapters, right? And these adapters are not always random, right? So if you had a lot more, then typically this would converge with each other. But here also, it doesn't look that bad. right? So the probability of having 80 GC on a given base pair after 12 to 13 bases is kind of random. So that is good, right? And again, you can see how the GC content is varying per sequence, right? Uh, okay, this is how much is the N content. So N is like, if it's not 80 GC, then it puts N, like something that it doesn't really know. So there was no N, so that's okay. And the length distribution. So as you can see that all the 1000 sequences are exactly of 76 base pair. And that's because when Illumina is giving the results, it only gives you 76 base pair. So it trims them. Yeah? So that's why all the 1000 out of 1000 are 76. Now this will depend on the kind of chemistry and the kind of sequencing you're using, because in some cases you might get different lengths, but in this case all of them are this way. Yeah. Then it tells you sequence duplication level. Yeah. So this is again because of adapter sequences. What it shows you is that if you take a single base pair, then how much is the duplication level? So like if you take one base pair like A, T, G, C, two would be like if you, let's say, take A, then how many times it is duplicated, AT, and so on, right? So there is a small peak here. That means there is a 10 mer. 10 mer would mean a sequence of 10 base pairs that has a slight duplication, right? So that might be some kind of adapters, and this is what you see in your overrepresented sequences. So in your overrepresented sequences, you will most of the time see some kind of adapters or some kind of indexing primers or such sort of things. So these are the sequences which are overrepresented. Yeah. Now the adapter, it shows that there is no adapter. Yeah, and that's because when we got the results, the adapter had already been removed. But maybe in your sequencing result, there are adapters, right? So this is just to give you a short summary of things that have happened. If you just want to look at this, the most important is this: per base sequencing content. After 12 to 13 base pairs, it should converge. So you should have it at a random probability for RNA-seq. Now, depends on what kind of sequencing you are doing. If you are doing chip-seq with something that binds to CpG island, then maybe it will be the G and C's will be overrepresented, right? So it depends. But for RNA-seq, generally, this is the most important one. If this is looking normal after 12 to 13 base pair, ignore the starting 10 base pairs. That means your RNA-seq more or less worked. Yeah. And also this. So these are the two ones: per base pair sequencing quality and per base sequencing content. Now, these are the two most important parameters that you need to look at. Yeah. Good. Yeah. I got confused with one of them. Sorry about that. Okay. Next, we want to remove two things. First thing is we want to remove the repeated sequences, the adapters, the indexes from the ends, right? And another thing that we want to remove are the bad quality reads. Yeah. Or at least part of the read which is not so good quality. So let's say if you have a 75 base pair read and only the first 30 base pair are of good quality and the remaining are not, then you want to remove it out, right? And this is what is called trimming. So trimming essentially accomplishes two steps: removal of adapters and removal of bad quality reads. Yeah. So that is the next step once you have your raw data, right? To do some kind of trimming. So again, we'll go back to tools. Yeah. And we'll go to FASTA/FASTQ. So this is about manipulating your files, right? And here, the tool to use is called Trim Galore. So Trim Galore is a tool for doing exactly what we want. It does both the things together. So here is Trim Galore, or you can also search for it here. Yeah. Essentially, it's running cut adapt, which is another tool in the background, but it does everything for you. So you choose Trim Galore. This is the next thing. Because you will be using this a lot, you can add it to your favorite. right? And then you just specify a few parameters and it will do everything else for you. So what you need to specify is whether your raw data is single end or paired end. Because if it is paired end, then generally you trim, no, not generally, you have to, if your data is paired end, then you have to trim both of your pair end sequences together. And why is that? The reason being, in paired end sequencing, uh, the position of the sequencing corresponds to the two ends, right? So the first read in both the files is one molecule, the second read is a second molecule, third read, and so on and so on. So just imagine if your fifth read in the first file is not good, and if it's removed, but the fifth read in the second file is good, and if it's kept, then this kind of correspondence is lost, right? And then you get confusion. So when it's a paired end read, you have to trim it together, and then what it does is, even if one read is bad, it throws away both the reads. Yeah, we'll go more into detail in the next session, but just to make sure that when you have paired end, you have to trim, you have to process them simultaneously, because there is a 1:1 correspondence between the two ends. Yeah, but in this case we take a paired end. Yeah, and because it's paired end, so it will be a single FASTQ file, right? One file. You can either it will come here, or we choose, but we have to choose here, right? Because no, sorry, we do it with this file. We have not done any processing on it, right? And the adapter sequence that has to be trimmed. Technically, we do not have to trim any adapter sequences, because our FastQC report told us that there were no adapters in these sequences. So we don't have to trim it, but we will just say automatically detect. Yeah. Otherwise, you can say user defined adapter sequence and just put empty. Yeah. And then there are a few other parameters, but you can use you can just put all of them as defaults, and you can also read about their description at the bottom. Yeah, the description of this tool, right? So we will execute it. So once you execute, of course, it enters into the pipeline. So it's the next thing that is going to happen. And another interesting thing that Galaxy does is that it shows you the top options of what other users do after executing this command. So after executing trim galore, these are the different things that people are doing. Yeah, and you will see that one of them is mapping. This is exactly what we will also be doing, right? So this is sometimes very interesting to look at. Yeah. So now our trim galore is running. While trim galore is running, I will show you two more tools which might be of interest to at least a few people I know. For example, Peer, if he's listening. And that tool is actually, if you have a single FASTQ file, right? So let's say you did an experiment, and you only did one replicate, right? But you want to do some kind of statistical analysis, like it was a preliminary experiment just to see, and you only did one replicate. Then there is a way in which what you can do is you can split your FASTQ file into two subfiles. So you randomly choose sequences, and then you call them as two technical replicates to do statistics. Yeah, so you can do a preliminary experiment with just one replicate and do statistics by splitting the file into multiple small files. Of course, when you publish it, it's not you will not be able to publish it like that, but sometimes you just do a preliminary experiment and you want to know what is happening, right? So you don't, if you don't have replicates, then it's almost impossible to do statistics, right? Because n equals 1, you cannot really do statistics. But you can split it, call them technical replicates, and then do your analysis, right? So how do you split it? So there is a tool called FA split. Yeah, and this does exactly the same thing. So FA split, very simple, it splits a FASTA file. Yeah, so you can choose it, And then you can say how many files do you want to split. You want to split this one file into, right? So let's say if you want to split your file into two, then you just type 2, right? I will not run this command because we don't want to do it, but if some of you are interested, or this is your problem, then you can do it like this, right? So the tool is called fsplit. Yeah. There is also another tool which is called subsample sequencing file. Yeah. This is if your file is very large, but you want to do your analysis a bit quickly. You just want to process it very fast, just get a preliminary idea. Then you can also use this tool, and then you can just randomly sequence. Let's say if you it tells you, okay, take every nth sequence. So that means which one do you want to choose. So if you say fifth, that means just keep every fifth sequence, right? So that means you decrease your file by 1/5, so you only have 20%, right? And you can read in which cases this could be helpful. Yeah. But the other tool is nice because it actually breaks them into multiple chunks, whereas in this case you can just reduce the coverage. But these are the two tools to reduce your file size if you want to either increase your technical replicate or you want to do your analysis a bit faster, right? Actually, this is the tool that I had used to get the first 1,000 reads to prepare the file for this session, right? So this was exactly the tool used to do that. Yeah. So just a couple of tools to have. Okay. So trim galore is finished. It has finished trimming. Yeah. And we will be able to see the result. Yeah. So we can click on the eye to view the data. And here is your data. So again, it just looks like a FASTA format, right? But what it would have done is it would have reduced some of the sequences because they had not such high quality scores. For example, you can automatically see right now the first sequence is bigger than the second one, right? And that means that the second one had some bad quality reads here, and this was trimmed away. So this is what trim galore is doing. If you want, you can run fastqc, so the fastqc quality control now on the trimmed data set. Yeah, so let's run that. So initially we had run fastqc on the raw data. Now we can again run it on the trim data to see what has changed between your raw data and the trim data, right? So it's just fastqc, but now the input is the item number 4 on your history. So you can also keep a track of this. So this is your item number 4, right? So item number 1, 2, 3, 4, right? Which is the result of the trim galore, right? So whenever you specify one of the things here, the data set, it's the result of that step. Yeah. Or when you browse, if you have done more and more analysis, all of these data will get added to your history and to your library, right? So we want to rerun this on the trim data set. Yeah. So now fastqc is running on this thing. So now while fastqc is running, we are ready to actually map the data. What does mapping mean? Because now you have removed your adapter sequences, you have removed your indexes, you have removed bad quality reads. So the only thing that remains is your own sequencing, the sequences that you are interested in, the sequences of cDNA created from the mRNA of your interest, right? So this is what it is. So now we will be doing mapping. And what is mapping? I think most of you must have done BLAST, right? So you take a sequence of DNA and you blast it against the whole genome or the whole transcriptome, right? So then the BLAST algorithm searches a match for your sequence against the whole against whatever you blast it with, right? And this is exactly what mapping does. It takes each and every one of your sequence and matches it, tries to find a similar sequence in the genome that you specify. So I think I have the slides are a little bit mixed up, but anyway. So there are 4 major mapping tools. Botai 2, BWA. These are these are now slightly old, but they were used a lot. The 2 new ones that I'm using most of the time are called HiSat2 and Star. Actually, on most of the clusters, people will be using Star. Yeah, so most of the time you will get your results from Star. Okay. The problem with Star is that it has very high requirements of RAM. Yeah. So it's a very intensive algorithm. So for mapping human, you need approximately like 32 GB of RAM to run Star, and that's why people in the like run it mostly on clusters. Yeah? Yeah, although if you have a computer with 32 GB of RAM, then you can use Star. Yeah. The other one that I really like is HiSat2 because it does not have that much RAM requirement. It takes a little bit longer. Of course, Star will be faster. HiSat2 takes a little bit longer, but you can easily run it like on a 8 GB, even 4 GB laptop. So it does not have problem. So for mapping, you will be mapping it against the genome, right? But as you can imagine, if you keep on taking each and every sequence and if you blast it, it's going to take a very, very, very long time, right? So you have to make it more efficient. The search algorithm has to be much more efficient. And the way the search algorithm is made more efficient is by using something that is called an index of a genome. So the index of the genome is to genome what a dictionary is to words, right? So if you want to find a specific word, like let's say definition in a dictionary, you just don't randomly go through pages, right? So you go to the region which has the alphabet D, and then to the region that has alphabet E, then F. So you do it in a more systematic manner, right? It's because in a dictionary all the words are arranged alphabetically. Similarly, in a genome index, it arranges your whole genome in k-mers. So k-mers is like 1 mer is 80 GC, 2 mer is 80 AA, so like 2 base pairs together, right? And it arranges it into k-mers. And then instead of searching for your sequence, you actually search for these short k-mers. So let's say you search for 8 base pairs together. And all of these 8 base pairs are then arranged just like in dictionary, they are arranged systematically, right? And along with their location on the genome, right? So this is one example. So for example, if you want to search the word banana, yeah, and you have split it into many small things. So let's say if your sequence is the full word banana, then directly it knows that it is at position number 0. It directly goes there. Whereas what you have sequenced is first an A, then an NA, then an NA. So your sequence is actually A, NA, NA. Yeah. Then it can still use the dictionary because it has split it into smaller things. It goes into this tree-like structure, and then it knows that it has to go to location number 1. Yeah. Whereas if your sequence is NA, NA, then it knows that it has to go to sequence number 2. Right. So essentially, index is a way of reducing the complexity to increase the efficiency of search. You don't have to understand how it's done. I have absolutely no idea how it's computationally done. The only thing that you need to know is that for each of the mapping tools, there is a unique index because every mapping is a unique algorithm. It's essentially a search algorithm. Yeah. So the index that you use is going to be unique to the mapping tool. And of course, it's going to be unique to the species and then unique to the version of the genome that you're using. Yeah. So every index will have 3 characteristics: the mapping tool, so which one of the 4 or any other one, which one of the mapping tool you are using, which organism genome you are using, and then which version you are using. For example, for human, are you using version 36, 37, 38, for zebrafish version 9, 10, 11. Yeah. So these three characteristics are important when you are doing your mapping. Yeah. So mapping is essentially you take your sequences and then you try to find the location in the genome. Yeah, that is mapping. So our fast QC on the trim data has finished, and if we are doing this in the live format, then we will keep doing it. So we'll run a command, go to something else, and then come back to it because it's going to take some time. But now let's see the result, and we are going to focus on two things. So first thing you can see is that the quality score at every base pair improved, right? So now it's not degrading so much, and that's because the sequences which did not have a good quality scores were removed, right? So this is what trimming does. Yeah, so the overall the quality score has improved. And the second thing is that you will see that there is some lack of complexity here. That's because it did not really find an adapter, and I think this is more to do with the fact that we only have 1,000 sequences. Yeah, but overall, anyway, at the end, it's kind of nice. Yeah, but another thing that would have changed, and I think you can imagine, is the length, right? The average lengths, and that's because some of the sequences would have gotten trimmed and some of the sequences would not have gotten trimmed. So sequence. Now there are some sequences. So initially, remember, all the sequences were 76 base pair, but now you have some sequences that are even like 50 base pairs, 70 base pairs, right? So in some sequences, you have had a lot of removal. But where was the length, the distribution of length? One second. Per base pair in continent per GC. Okay, here it is also represented. So the sequence length now varies from 22 to 76 base pair. Yeah. So in some of the cases, and also you see that two sequences have been completely removed. So they were crap, right? So 2 out of 1,000 were completely crap. They have been removed. Yeah. So this is what trimming does. Many of the time you can get away with not trimming, but it's a good thing to do, and for sure if you want to publish something, I would strongly suggest you to trim it. Many times your result doesn't change by a lot by not trimming, because the noise that will be in one of your sample may also appear in another one, but it's always a good idea to trim, and it also doesn't take that much time to do it. Yeah. So now the next step is mapping, right? We are going to take the output of the trim data and find the sequences in the genome where they align to. So where will that be? Yeah. Multiple alignments. No, let's just search for it. So we will use HiSat2. Yeah, so HiSat2, fast and sensitive alignment program. Yeah, and this is going to take a lot of time. Yeah, so I think we won't be able to see the remaining results, but when it finishes for you, then you can look at them. And this will be one advantage of Galaxy, is that you can already execute commands even if you do not have the results, expecting that you will have the results in future. Yeah, so we will put all the commands, and then once they finish, they will finish. Yeah. So first of all, you need a reference genome. So which animal, which organism, and which genome do you want to map your data to? So this is the zebrafish data, so we'll use zebrafish. Yeah, and we'll use built-in genome. So here we will search for zebrafish. Yeah? And we'll use the Danner 11, Danuario 11. the most recent one. Something that is important to realize is that Galaxy is connected to UCSC. So it is not using the genome from Ensemble, but it's using the ones from UCSC. And this is important because even the genomes between Ensemble and UCSC are different, and their corresponding transcriptome, right? So therefore, whenever you want to refer to any location or anything, and if you're using Galaxy, you have to go to UCSC website, yeah, not to the Ensemble website. Yeah. And then you have to specify what kind of library it is. If it's paired end, then paired. Then you will have to specify 2 files, but today it's single end, and you specify which one. So it will be the output of the trim, right? Of the trim Galore program, which will be the trimmed reads, right? And the data should be either FASTQ Sanger or FASTA. So either FASTA format or FASTQ. Yeah, Galaxy just calls FASTQ as FASTQ Sanger. And you can see that the output format of trim Galore is actually FASTQ Sanger. Yeah. And this kind of sequencing was done unstranded. So that means there was not a preference for the strand. So if you are using random hexamers, then most of the time it's unstranded, but you can always ask the person who did the sequencing or the platform that did the sequencing about the strand. Yeah, but in this case it was unstranded. So this is an information that you'll most possibly get from the platform. Yeah. And then you just say execute. If you do not know, you can put unstranded. It's not a big problem. Yeah. And then you just execute it, right? Now, after you execute it, you will see that it says that the output will be BAM, BAM, right? Now, what is the BAM format? What kind of output are we expecting? The job is already running. Hopefully, we will see it will finish. So, once you have aligned your file, your reads, you will get the output in 2 different formats. One is the SAM format, and the another one is the BAM format. SAM format is essentially full form for sequence alignment map or format, right? And this is human readable. So you can actually open it and read it. And essentially a SAM format is very simple. It's your full FASTQ data, so your sequence data and your quality data, along with the information on alignment, along with where that read mapped onto the genome, and some more metadata. Yeah. So essentially the SAM file will at the end of the day look like this. So for each read, you will have one line. Yeah. And there will be the name of the read, which is the header in FASTA format. Then there are a few other things which are not of use. I mean, the position on the chromosome and all that, all those things. And these two are again coming from your FASTQ file. So everything that is blue here is coming from your FASTQ file, the read name, the read sequence, and the metadata. So it doesn't lose any information, it adds to it. And that is why SAM files are very big. Typically almost double of your FASTQ file. So if your FASTQ file is, let's say, 5 GB, then SAM file might reach 10 GB, right? Because it's going to add information onto your FASTQ file. It's not going to shrink it, right? Also, if you have paired end reads, then it will be double of the total size of your 2 paired end, right? So if each of your paired end is 10 GB, then the SAM file might actually even reach like 14, 15 GB. Yeah, so they are very big. And that is why to save, just an example of how SAM file will look exactly, because Galaxy will not give a SAM file because they are too big, is that this will be the header for the SAM, and it will have some information on which kind of mapping was used, what genome was used, and so on and so on. And then for each line will have information about your read and the mapping location. So for example, this is the read, this is the quality score for each base pair, this was the name of the read, and then it mapped to chromosome 5 in this location. Yeah. So these kind of things. But Galaxy, and also a better way to save this alignment data is in BAM files. And BAM files are essentially compressed SAM files. So just like gunzip is a compressed fastq file, right? Similarly, it's an algorithm, a compression algorithm for the SAM file format. And because they are compressed, so they are not human readable. Just like you cannot really read zip files without opening it, right? So typically the size decreases 1/5, so 20%, and that's why they are more easier to store for storage purposes, but they are not human readable. But anyway, you won't even need to read them. Yeah. Okay. Now let's see if it has. Oh, okay, it finished. That's good. I was not expecting that it would do it so fast. Okay. So now you can see that your result is 138.6 KB. Let's see how much did we start with. If it shows the file size. Okay, let's go to the original one. The original one was, do we have our data sets? Let's see our data set. Just how much? So it was 77.5 KB, the original data set. And the BAM file is 138.6 KB, right? So even the BAM file is bigger. So the SAM file will be 5 times. So it would have reached 1 MB, right? Approximately. Yeah. So now what does it show you? This is how exactly the BAM file is looking, right, at the top. So Galaxy makes it human readable, right? So this is the top header. Yeah. And I think after a while you should start seeing some sequences. Yes. So here now you start seeing your sequences, right? So this one has mapped to chromosome 5 and so on and so on, right? So now you have found the location of each sequence in your genome. Now to visualize it, there is a very nice option here, display at UCSC. So this will, what it will do is it will take your BAMP file and put it into the UCSC genome browser with the genome that you used. So if you click on it, we only have 1,000 sequences, so we might not be able to see much, right? But you can always do that, right? So this right at the top are your reads. So if there were like millions of read, you will see peaks here. Yeah. Now we sequence. So this file is zebrafish beta cells. So beta cells express insulin. I just know. I mean that. So we will go to the location of insulin. Yeah. And here you see that you do see that a few of the reads, like even out of these 1,000, we do see a few of the reads being aligned here. Yeah. And this is the insulin gene. So this is, so another thing is that this is RNA-seq, right? So we should mostly be seeing exons, and that is what you see, right? So this is an exon, we have a read here, or at least the reads cover this area. This is an exon, the reads cover this area. This is an intron, the reads do not cover this area, right? So you have the exon as well as the 3' UTR, and the reads cover this area. So it looks good from an RNA-seq perspective. It looks like it looks like RNA-seq. Yeah. So we have done our mapping. We know the location of each reads in the genome, but we only know it on the genome. We do not know how many reads correspond to a single gene, right? So we have not yet done counting. Yeah, we have not yet counted the read per gene. So that is the next step that we have to do. We have to do counting, right? And for counting, we will be using the tool called HT-seq count. So you can search for it. It's under RNA analysis. Either you can search for it or you can go and look at it here, RNA-seq analysis, sorry, RNA analysis. And it is called HT-seq count. Yeah. I'll just search for it, it's easier. So what this tool does is it takes a BAM file and it takes a GFF file, right? And it will give you the counts. Now, what is a GFF file? So first we will look at that. So the genome has information about what are the sequences on each chromosome, right? But it does not really have information about how they are distributed or how they are aligned in terms of genes. And this information is given in this kind of file format, GTF or GFF3. Yeah. So in this file format, it's a simple text file which is separated by tab. So it's a tab separated file in which each column has some sort of information. So it has the chromosome location, it has the name, right? It has what kind of sequence is there, where does it start, where does it end, what's the strand, so positive strand, negative strand, and then the ID. So it could be ensemble ID, it could be the gene name and such sort of things. Yeah. But it's very important to note that the format of GTF that you are using, it's compatible with uh, the genome that you used. Yeah. Because for every genome, you have a specific GTF file, right? So for example, the GTF file for from ensemble will not work with UCSC, yeah, and vice versa, right? So this is something to be careful about, yeah. And I think so we will go and search for the GTF file, yeah. But before that, just give me 2 minutes because there is somebody at the door, so I will just come back. Yeah. Hello. So sorry, we had some plans for Friday evening. So I guess we'll do it for 10 more minutes, yeah, and then we can be done. Yeah. Sorry about that. Sorry for the interruption. Yeah. So we need the GTF file uh, for zebrafish, right? And because we used Galaxy, which uses UCSC genome, we have to get our transcriptome data from UCSC. So first we go to UCSC browser. And then we go to downloads, genome data. Yeah. And here it will show you all the organisms and the genome data that you can download. Yeah. So we are using zebrafish, right? So we go to UCSC browser, we go to download genome data, and then to zebrafish, right? So it's very important that you use the exact same transcriptome for which you had used the index of the genome, right? Otherwise, the chromosome locations will not match, the chromosome name will not match, and so on and so on, and then it's not going to work, right? So we had used this one from UCSC, Dandrer 11, from May 2017, right? We can go back and check. So we can go to info here, right? And here it says Dandrer 11, right? So that is why we have to use the same transcriptome. So this is something that's very important. Yeah. So we go here, we go to UCSC, we go to the genome data to download, and here is all the different kinds of data. I'm going to zoom in. Yeah. So this is actually the genome. So here, danrer11.fa.gz is the full zebrafish genome. And similarly, you can find the same thing for human, mouse, everything. Yeah. You cannot use the genome directly because you'll have to first make an index. Yeah. And this index, the Galaxy people have already made. So this you don't have to worry about. But the genes are here. And this is your transcriptome data. So your different GTF files. Yeah. You can download any one you want. All three will work. Yeah. So I will just download the ref gene. You can also download all three, and in Galaxy run all three in parallel. It doesn't matter, right? And you can see how the results differ. Yeah. So we will download the ref gene file. Okay. We will say save it. And now you have the zebrafish transcriptome data. So again, just like last time, we want to upload it. So we go to our FTP client. Either you can go to the FTP client, or you can go to you can directly download it from your computer. So upload from computer, choose local file. It's not so big, so it will get uploaded, but we will just do it in here in the FTP client. So we say upload and the GTF file. Good. So the GTF file is uploaded. Another important thing that I forgot to mention, I think, is that HTC count needs GTF format, not GFF3. And what the difference is just very minor. It's just depending on what each column represents. So this is the only difference between GTF and GFF3, just what each column represents. HTC requires a GTF format. Yeah. So these two are not different in any other way. They both represent transcriptome. The only difference is what each column represents. Yeah. Okay. So we will go to the FTP site, and this time we will say that I want to upload my transcriptome. I close that. Yeah. And so it's going to take some time uploading. It doesn't matter. You can already go to next step. So we go to HTC count. Yeah. Now it needs two data sets. It needs a BAM file or I mean, if you run it yourself, you can give it SAM or BAM, it doesn't matter. And then it uses a transcriptome file. So now since your transcriptome file is uploading, you can already choose it. This is an advantage of Galaxy. You don't have to wait for the execution to finish. You can already make your pipeline. Yeah. Everything else you can keep same except the stranded, right? Because our data is not stranded. Yeah. And everything else you just keep normally. Yeah. You can read about what each parameter means down here. So it explains every step of it, but you just run it with default, just taking care which kind of strandedness there is. Yeah. So we can say execute and then look at our transcriptome file. So this was the uploaded transcriptome file. And when we say view, we see how the transcriptome file looks, right? So the first column is the chromosome. The second column is just the source, where it comes from. The third column is what kind of characteristic is there, the start and end, the strand. If it's an exon, then the frame, and then its attributes. So all the attributes are in one column. So these attributes are gene ID, transcript ID, and so on and so on. Now, one thing that you can do is, remember here, I will just do it again. When we did HTC seek, we said no, and then I said to just choose default. In there, one of the attributes was the ID attribute. So which ID do you want to compile? Do you want to have information for gene? So what are all the counts per gene? You can also have it for per transcriptome, right? So if you go to your GTF file and you look into the attribute column, then you will see different features. You will see gene ID, you will see transcriptome ID, and you may see gene name, gene symbol, so on and so on. You have to specify which one do you want to use to aggregate. So gene ID will give you for gene, but we could also do it. So here, we could also do it by gene name, right? Because gene name is another attribute, in present in this GTF file. We could also do it for transcript. And if we did it for transcript, then you would have your data with counts per transcript, right? So this is another attribute that you can change. Right? So the two things that for HTC count you need are BAMP file and the transcriptome file, and the two things that you might want to think about is the strandedness and the attribute that you want to aggregate for. Yeah. And the HTC count has finished. So it gives you again two kinds of results. One is just the overall score. Yeah. So it tells you that it could not align 268 sequences. So 268 sequences were not present in the transcriptome. So maybe they were either repeats or something there. Yeah. It also says that 550 were not aligned uniquely, which is okay. It's not such a bad deal. Uniquely means that it found multiple locations where it could align, but it can happen. And then 195 were aligned, but they were part of not a feature. So they were not part of exons or UTRs or such sort of things. Yeah. So the remaining were aligned. So if you want to look, then you would have 998. That's what we started at. 998 minus 268 would have aligned. So to look at the result is here. Right. So the first result for HTC count. So HTC count gave two results. The first result is oh, hmm, is the counts, and the second result is the summary of the whole thing. Yeah. So here you will see each gene ID. In this case, here are the genes. So for each gene ID and the counts. And because we only had 1,000 counts, so it's very less. But one thing you can do is search for insulin, INS, right? Because that's a gene that's present. And you see that you get 3. So that means out of 1,000, 3 cDNAs map to the insulin gene, somewhere on the insulin gene. Yeah, it could be an exon, UTR, anywhere. Yeah. And that is it. So now you have gone from your raw data to counts. So now whenever you get your raw data, at least you should be able to get to have a knowledge about what is the proportion of reads mapping to each gene. Yeah, and then the next thing is to analyze the data, right? So next time we will start with how to make this into an automation. So how to automate this whole process. So one thing that I would advise you to do is if you are going to work with something else, to make a new history, because one good advantage is that if you have done any kind of analysis, the whole analysis gets saved as a history, and you can go back and forth. So if you have multiple histories, you can switch between them. So like for example, this was the history that I practiced for today's session, and so it's saved as one thing. And this is what we did today, so it's saved as another thing. So if you make another history, then you can always keep on switching back and forth. So as in history, you will have everything saved. And so next time we will make it more, we will look at how to automate it, what to do with paired and read, and how to analyze the RNA-seq dataset. Okay, so that's it. Thank you for your attention, and hope you liked it. and we'll see each other in 2 weeks. Yeah, not next week, but in 2 weeks. Thank you. Enjoy your weekend. Bye bye.